Hello, everyone, and welcome to today's live broadcast, From Cell Seeding to Analysis, Getting the Best Out of Your Cell-Based Assay, presented by Dr. Jessica Wagener, Application Specialist, Cell Handling, Eppendorf AG, and Dr. Erik Bonke, Application Specialist, Promega, Germany. I am Alexis Krauss of LabRoots, and I'll be your moderator for today's event. Today's educational web seminar is brought to you by LabRoots and sponsored by Eppendorf. For more information on our sponsor, please visit eppendorf.com. Now, let's get started. Before we begin, I would like to remind everyone that this event is interactive. We encourage you to participate by submitting as many questions as you want at any time you want during the presentation. To do so, simply type them into the Ask a Question box and click on the Send button. We'll answer as many questions as we have time for at the end of the presentation. If you have trouble seeing or hearing the presentation, click on the Support tab found at the top right of the presentation window, or report your problem by clicking on the Answer a Question box located on the far left of your screen. This presentation is educational and thus offers continuing education credits. Please click on the Continuing Education Credits tab located at the top right of the presentation window and follow the process to obtain your credits. I'd like to now introduce our presenters, Dr. Jessica Wagener and Dr. Erik Bonke. For a complete biography on Jessica and Erik, please visit the Biography tab at the top of your screen. Jessica, you may now begin your presentation.
Thank you, Alexis, for the introduction. Welcome, everyone, also from my side, and thank you for joining the webinar today. I want to start by having a look at the contents for today's presentation. First, I will talk about the general aspects of cell culture routine and how to increase reproducibility of your data when preparing the cells for an assay. And then Erik will take over the next part and go into detail about optimizing assay setups. And in the end, I will touch the topic of how to avoid cryptic contamination that can influence your assay results. Here's an overview of the complete cell culture workflow. So independent of the specific cell type you're working with, these steps are part of your lab routine. For this webinar, Eppendorf and Promega are joining forces to optimize your cell-based assay results. We will cover the general aspects like optimal seeding techniques and cultivation conditions and go into the details of different modes of detections and assay setup. But before we proceed, here comes our first poll question. We are interested in the cell types you're working with. So the question is, what kind of cells are you using? And you can select all that applies. So the answers are continuous cell lines, stem cells, or other primary cells. And I will give you a few seconds to vote, and then we will share the results with you. So please click on the results now. Okay, so the answers are coming in. We're going to push the results now for you. So we see that the majority of you, more than 80%, is using continuous cell lines. 10%, about 10% is using stem cells, and almost 14% is using other kinds of primary cells. Okay, so no matter which cell types you are working with, there are some aspects that can be considered to increase the reproducibility and consistency of your assays or your experiments in general. And this is what we are looking at in the next chapter. Okay, so let's have a closer look at this first experimental step that is so important, the cell seeding. The preconditions for getting comparable results is to seed out the same number of cells for every negative control, positive control, and for every experimental replicate. The more replicates you include, the better for your statistical analysis, of course. Variations in seeding cell numbers and the formation of air bubbles during seeding, especially when you work with small 96 wells or smaller wells, during seeding will not only increase standard deviations, it will also make your results less reliable. When you fill, for instance, a multi-well plate with cells, it will take some time until you have filled all the wells of a plate. So you can use a single-channel pipette and have the cell suspension in a tube, or you can use a multi-channel pipette and have the cell suspension in some type of reservoir. No matter which tool you use, the longer the seeding process, the higher is the variation in cell number from one well to the other. As you see in this image here on the upper right, the cells sink towards the bottom of the tube with time, and this sedimentation goes quite fast, within minutes. Therefore, you need to resuspend or mix the cells to swirl them up in between to get an equal number of cells in every well. Saying that, we should keep in mind that cell culture media tend to foam due to the high protein content of supplemented serum. Air bubbles can hinder cell attachment. In the lower image shown here, you can see how an air bubble hinders the cells nearby to adhere properly to the surface. So you should avoid harsh and fast pipetting to reduce the formation of air bubbles and also to reduce shear forces which further stresses the cells. In fact, there are a lot of aspects about pipetting that are often underestimated. Pipetting is an everyday task and can have a big impact on cell culture experiments. Not only the type of liquid influences the pipetting results, also the pipetting technique plays a role. So when aspirating the liquid, hold the pipette vertical, as seen here on the left image, and immerse the pipette tip only slightly and try to keep a constant immersion depth. If you follow this technique, you will improve the precision of the volume that you are pipetting. Now let's have a look how pipetting can affect a cell-based assay. In this graph on the left, you see the results of a cell viability assay. Each column represents one row of a 96-well plate. The aim was to seed an identical number of cells in each well. 2 rows were left out for blanks here. The cells were seeded with a manual 8-channel pipette from a reservoir. After 24 hours, the number of viable cells was determined using a colorimetric assay. And in rows 1 to 5, you see varying cell numbers in the wells and therefore high standard deviations. Here, the user did not mix the cell suspension between pipetting each row, so the cells sedimented in the reservoir with time, and in addition, the pipette was held in different angles and heights during aspiration. And in rows 6 to 10, you see the results with mixing the remaining cells before pipetting each row, aspirating at constant height and in upright position. So taken together, these data show that the pipetting technique has a big influence on the results. Besides the technique and the pipetting tool, the other thing that can affect results of an assay are so-called plate position effects. If you work with a 96-well plate, the usual volume you use is 100 to 200 microliters per well. Especially during longer incubation periods, evaporation can become a critical factor with a small volume, especially in the outer wells, because here the wells are not surrounded by other wells with warm liquid inside. The evaporation becomes more severe. So this is a phenomenon that we call the edge effect. So the outer wells of the plates cannot be used because the evaporation leads to different cellular responses. And what you can do to avoid this edge effect is to fill the outer wells only with medium or PBS to insulate the inner wells. If you do that, you can avoid this edge effect, but you reduce the number of usable wells to 60, which is a loss in capacity of 38% per plate. The Eppendorf cell culture plate has a surrounding moat that can be filled with liquid. Thus, the outer wells get insulated and the evaporation effects are minimized. And here we measured how effectively the edge effect is reduced in the Eppendorf plate. Without insulation, the evaporation in standard cell culture plates can go up to 15, almost 20%, as you can see here in the upper left image. And again, if you think about a volume of 100 to 200 microliters per well and you want to test maybe different concentration of a substance, over time the evaporation will influence the concentration of the substance and the cellular responses are not comparable anymore. In the graph here on the lower right, you see how the different evaporation rates can affect cell proliferation over time. So with insulating the outer walls, you ensure stable conditions throughout the complete plate. In addition to stable conditions throughout the plate, the conditions for the cells in the CO2 incubator should be as stable as possible. Every time the door of the incubator is opened, the atmosphere inside is disturbed. Temperature and CO2 level all influence the cells. It's a good advice to reduce the traffic in and out of the incubator as much as possible. Only open the door when necessary and close it as soon as possible. Some incubators show temperature overshoot to reduce the recovery time after door opening. This can harm the cells, especially when you work with sensitive cells. So your incubator should have a quick recovery of CO2 and temperature without any overshoot, as seen here on the upper right graph. Especially when you work with very sensitive cells, or when you work in a lab where many people are sharing an incubator, it can be a good idea to have separate incubators for maintenance cultures and running experiments. So you seed the cells for your assay, do the treatment, manipulation, or whatever you want to do, and put the cells into a separate incubator for the time of the experiment. Your colleague who has to split the cells or change the medium uses the other incubator, and thus your experiment is not disturbed by fluctuations of the inner atmosphere. Here on the lower right, you see a screenshot of the menu of the new Eppendorf Cell Expert CO2 incubator. The red arrow here indicates a door opening, and you see that the curves of CO2 and temperature are dropping. Monitoring cultivation parameters and documenting performance of your incubator are measures to establish long-term reproducibility of the experiments and assays in your lab. You can simply check for door openings or track back the performance data to see what might have influenced your assay this time. So before you start the maybe more complicated troubleshooting of the assay itself, it might be worth looking into the performance of your incubator. Let's have a closer look at the cells in culture. So no matter what you do with a cell culture, it is important to determine the right time point for a particular cell type or cell line. In this graphic here, you see a typical growth curve of cells in culture. It is important to not let the cells overgrow until they are in the stationary phase and do not proliferate anymore. So the optimal time point is when the cells are in their exponential growth phase, or also called log phase, and this is when they are between 70 and 80% confluent. Plotting a cell growth curve and determining the population doubling time for a cell line is critical for understanding the optimal seeding density and the optimal cell concentration range for the routine subculturing of a cell line. And when you launch an experiment, it is important that you can be sure that any changes in cellular behavior are a direct response to an experimental stimulus. For example, you want to test how the cells react to the treatment with a certain substance. So you add the substance to your cells and see increased cell death, or the proliferation rate slows down, for example. So now to evaluate a direct correlation between the changes in your cell culture and the experiment you performed, a constant culture routine is important. So a growth curve provides information about behavior during cultivation and thus facilitates the assessment whether any biological response can be attributed to the effect of a tested factor or whether it may reflect an imbalance within the culture system. Another factor that has often been described to influence cellular behavior is the passage number. The passage number is a record of the number of times a cell culture has been subcultivated. There are numerous studies giving evidence that the passage number affects a cell line's characteristics over time. It has been described that cell lines at high passage numbers experience alterations in morphology, response to stimuli, growth rates, protein expression, transfection efficiency, and more. So looking at all these passage number effects in different cell types described in the literature, the basic question one can think of is, how many passages are too many? And before we go on, I want to hand over this question to you and start our next poll. So what do you think? How many passages can a cell line be cultured for? And the answers you can click on are not more than 20 passages, 20 to 60 passages, up to 100 passages, or the passage number has no influence on the cells. So again, we will wait a few seconds and give you the time to answer until we are showing the results to you. Okay, so the answers are start to coming in now. All right, so we wait a couple of seconds more. Okay, so we are pushing the results, so you should now see the results for this question. Okay, so about 53% of you thinks that not more than 20 passages is okay. Around 36% is using their cells for 20 to 60 passages. Only 11% is using up to 100 passages. And there's no one who clicked on the last answer here, so no one of you thinks that the passage number has no influence on the cells. All right, thank you for participating. And so, in general, we can say keep the passage number as low as possible. Especially primary cells that have a finite lifespan are prone to genotypic and phenotypic changes as they adapt to in vitro culture. After a number of population doublings, they will stop growing. In contrast, continuous cell lines like cancer cells or cancer cell lines have an unlimited lifespan, but although they can proliferate indefinitely, also their genotype and phenotype can change over time. A lot of the well-established and well-characterized cell lines that are used in laboratories worldwide have been isolated from their original tissues decades ago. So when you work with a continuous cell line that has been isolated from its original tissue like 40 or 50 years ago, it is most likely not possible to obtain early passages from the cell line, of course. But you have to set a starting point as a reference to be able to obtain reliable results with this cell line. First of all, the starting point should be cells of high quality. The best way to ensure this high quality is obtaining cells from one of the established cell banks, like the ATCC in the United States, the ECACC in the UK, or the German DSMZ. Here you get well-documented, authenticated cell lines which serve as the best reference culture for your experiments. By establishing a reference cell stock, you have a constant supply of early passages from your cell lines. Now you can determine the safe passage number for your cell line of interest by establishing certain baselines. For example, test the cells for the presence and expression level of a certain protein or marker, monitor the proliferation rate over time, keep an eye on any changes in cellular morphology. By getting to know your cells, you will get an idea of whether and how the cells change in relation to prolonged passaging. And maybe a very simple advice, do not keep cells in culture that are not actively used for your assays. So don't passage continuously, but rather establish new cells from frozen stock if required. And now I would like to hand over to Erik from Promega, who will go into detail about optimizing the setup of your cell-based assay.
Thank you very much, Jessica. And also thanks everyone for joining this webinar. So on this section of the webinar, I would like to talk a little bit about how these cells that we've learned how to ideally handle them can actually be used for in a, so to say, experimental setting, a so-called cell-based assay. And primarily, I would like to focus on different key factors which are known to significantly influence the outcome of such an assay. First of all, I like to talk a little bit about the most frequently used modes of detection regarding cell-based assays, and also then related to this about choosing the optimal or compatible microtiter plate. I then would like to speak a little bit about how the actual design of your assay, as well as the cells themselves, and finally also the readout, so what you measure with the assay, and last but not least, the timing of your assay can affect the result of your experiment. If we have a brief look on what different modes of detections are frequently used for cell-based assays, the first option you have is an absorbance-based assay. In an absorbance-based assay, you have a light source, and the light produced by that source travels through a sample, and what you determine is basically the decrease in signal intensity due to absorption of light by components within that sample. The second option you have is a fluorescence-based assay, and again here also we have that external light source, which is an excitation source that produces light of a distinct wavelength, which we also know by the excitation light. And if that light hits a fluorophore within the sample, the fluorophore will absorb that energy. It will in turn emit some of that energy in form of light, so we have an emission of light at a longer wavelength. And the third option you have is a bioluminescence-based assay. And in contrast to the other two assays, in this assay we have no external excitation source. However, the light that we detect with our assay is actually produced directly within the sample in frame of a continuously occurring chemical reaction. If we now compare the attributes of these different detection modes, we can nicely see that, in fact, absorbance-based assays suffer from a very low sensitivity, low signal-to-background ratio, and a low dynamic range. These parameters are kind of improved in a fluorescent-based assay, however, you will get the highest sensitivity in a bioluminescence assay. And the reason for this is mainly due to the high background that is observed in a fluorescent-based assay due to a phenomenon that we know by autofluorescence. And due to that high background, of course, we have a decrease in signal-to-background ratio, and also the dynamic range is narrowed. And if we look at a bioluminescence assay, we almost have no background, and this is in fact the reason why we can achieve these high signal-to-background ratios and also this wide dynamic range of usually around 8 to 9 log units. Also, another disadvantage of absorbance-based assays is that there are, in general, not applicable to multiplexing approaches, while the fluorescence and bioluminescence assays can be easily combined in the multiplexing approach. Multiplexing means that you detect two different parameters using the same sample by application of two different assays that you subsequently perform. Now that you made up your mind what detection mode you want to go for, you surely have to select the correct plate type which is compatible with that distinct detection mode. In general, for a cell-based assay, you want that plate to be a tissue culture treated sterile plate, and you also want it to have a lid to allow for incubation. However, then we can distinguish three different plate types. So we have clear plates, black and white plates, and the black and white plates are also available as solid and clear bottom plates. The advantage of the clear bottom is that you can visually inspect your cells using a microscope. However, the disadvantage is that you will lose some signal, at least to some extent. If we perform an absorbance-based assay, we usually use a clear plate, and depending on what region of the spectrum you're looking at, you can choose from different materials. What I want to convey is that you should never use a clear plate for a luminescence assay. And the reason for this is that you have in a luminescence-based assay a continuous production of light, and this light in a clear plate can travel all the way across that plate, so you would not be able to track that light to a distinct well within that plate. if we perform a fluorescence-based assay, the primary choice is a black plate. And in contrast to what is usually stated that this is due to a decrease in crosstalk, the actual reason for that black pigment which is added to the plastic is that this pigment decreases the autofluorescence of the plastic, so the autofluorescence by the plate itself. And this, of course, decreases the background in your assay. The primary choice of plate for a luminescence-based assay is a white plate. And in a white plate, a brightener, titanium dioxide, is added to improve the reflective properties of these plates. And due to this, more of your signal will be reflected towards your detector, so you could say you kind of have an amplification of your signal due to that white color. And also in a luminescent-based approach, this white color decreases the well-to-well crosstalk. If you use a white assay plate, you should be aware of a phenomenon known as phosphorescence. So it's probably the worst place to store your plates on the window bench exposed to sunlight, because then actually some part of your signal will be related to the plate itself due to that phosphorescence phenomenon. So we recommend to store these plates in the dark, or to at least give them some time at the dark prior to performing that measurement. On the next slide here, I would also like to show you that there are differences in qualities, so a white plate does not unambiguously equal to a white plate. And this has been shown here by that very simple experiment where we compared two different plates and we performed a serial dilution of a luciferase in that first column of that plate, and we filled the rest of that plate with phosphate buffered saline. What you can nicely see is that in plate A, the signal generated in that first column can actually travel all the way across that plate. So we have a very high crosstalk, while in plate B, the signal is mainly restricted to the site of its origin. And also, if we compare the absolute signal intensities, these are way higher in plate B when compared to plate A. And this shows us, or at least suggests, that the reflective properties of plate B are higher than in plate A, potentially due to the addition of a higher amount of that brightener during the manufacturing process. Nevertheless, plate B has the properties that you really want to have in a cell-based assay, because you have a minimal crosstalk and a high signal output. Another factor that you might want to consider when setting up a cell-based assay is that you really want to reduce the total amount of steps required to perform that assay, because every step you add can be a putative source of error. And this is also the reason why actually all Promega assays are based on a fundamental principle, the so-called add, mix, and measure principle. And this principle means that we have a single reagent which we only need to add to our cells, we briefly mix, and then we can immediately perform the measurement. So due to this reduction in pipetting steps, we not only simplify the assay for the operator, but also make it more time convenient. And again, we significantly reduce the error sources and therefore increase reproducibility of the assay. Another factor that I would like to only briefly touch is the edge effect that we've already heard of, and which, as we learned, mainly affects the outer well of a microtiter plate. And this effect becomes actually apparent on two different levels. So first of all, in the outer wells, we have a higher degree of evaporation of the medium, and therefore we have a concentration effect of salts that are within that medium. And this concentration effect can cause changes in cell growth and cell viability. As a measure to counteract this effect, we can use the specialized plates that we've heard of, or we can only use the inner wells of that microtiter plate and fill the outer wells with pre-equilibrated sterile water or PBS or medium. And the second effect or level that this effect becomes apparent on is a temperature effect, because you can imagine that the outer wells of such a plate will be the wells that respond the most sensitive towards changes in temperature, which, for example, occur when you shuttle that plate from the incubator to your reading instrument. So our recommendation is that you really keep temperatures constant over time. For this, you could also use your reading instrument as an incubator, at least if it has a heating function or potentially also an atmospheric control. Nevertheless, you should always pre-equilibrate all the reagents that you use, as well as the reader, to the temperature that you actually want to perform the measurement at. Another source of variability in your assay is the seeding density of your cells. And this is shown by this experiment where we took cells from a stock culture flask and we seeded them at different densities, low, medium, and high. And then these cells were treated with a toxic compound, and two parameters, the ATP content and caspase 3,7 activity, were monitored. What you can nicely see is that the response of these cells to the respective treatment really differs. So apparently the cells seeded at the lowest density respond more sensitive towards the treatment with tamoxifen than the others. And additionally, not only the seeding density in the microtiter plate, but also the seeding density of your cells in your stock culture flask can have an effect on the response of your cells towards the treatment. And this is shown in this experiment where we used 3 different stock culture flasks at 3 different cell densities, low, medium, and high, and we used these flasks to seed cells at an equal density in microtiter plates, again performed a treatment and assayed the same 2 parameters. And what you can see that again the cells seeded at the lowest density or from the stock culture flask of the lowest density respond more sensitive towards the treatment with vinblastine than the others. So our recommendation is that you really implement standard operating procedures in your lab regarding your cell culture to make sure that you have fixed time points and fixed cell densities that you use in order to decrease this variability source. Another factor that may be a variability source in your assay, which is also related to your cells, is the metabolic state of your cells. As you can see by this experiment here, where we measured the consumption of the 2 commonly added substrates, glucose and glutamine, these substrates actually deplete relatively fast, and on day 2, so after 48 hours, they're actually already nearly gone. So at that time point, when the substrates become limiting for cells, the cells have to adapt and they undergo a metabolic switch. So if you use these cells then for your assay, this might cause some sort of variability. And also going along with a decrease in the substrate is an accumulation of metabolic products such as lactate and glutamate. And the accumulation of lactate is what we recognize by a change of the medium color from red to yellow. And this acidification of medium can in fact cause changes in the viability of your cells. So it's probably not a good idea to culture yourselves over the weekend, and on Monday, if you discover a yellow flask, to use these cells then to perform your assay. But also changes in pH in the other direction, so in the basic region, due to an increase in pH, can cause stress for your cells. And this is something that most frequently happens if you leave your media bottle in an open state under the bench for a long time, because then your media will become more basic because you have that low concentration of CO2 in the atmosphere. So as you see, cell viability is really a key factor in reducing data variability, and you have to perform cell culture in order to really maintain a high viability. Viability assays allow you also to optimize your cell culture procedures for a given cell line and to really establish a protocol that preserves a high viability. So you can use viability assays, for example, to compare different culture media or the effect of different batches of FCS. You can compare different trypsinization times or centrifugation times, centrifugation forces, so that you really optimize your cell culture procedure to ensure that you work with highly viable cells. Common markers that are used to check for viability are reduction equivalence. You can monitor ATP content or DNA content, which changes due to the division of cells. You can look at the membrane potential or measure the activity of specific enzymes. In contrast to checking cell viability, you could also check cytotoxicity, which then is a measure for the relative amount of dead cells in your cell population. And depending on what marker you choose, you can then also conclude on the respective mechanism that these cells have died from. Common markers are membrane integrity, which gets lost during cytotoxic processes, the release of lactate dehydrogenase, protease activities can be determined, membrane composition can change during cytotoxicity, and also the decrease in ATP as well as NADH content is indicative for cytotoxicity. The reaction that you can see on this slide is the reaction of the Firefly luciferase, which is fundamental to most of Promega's cell-based assays, and it is actually the conversion of the substrate luciferin into oxyluciferin and light, and for this the enzyme needs different cofactors, ATP, oxygen, and magnesium, and we do not use the wild-type luciferase because this is an enzyme with low stability. We use a modified recombinant version, which we call the UltraGlo recombinant luciferase, which has a higher stability when compared to wild-type. However, if you limit a distinct component of that reaction, you can now, for example, quantify gene expression. If you limit the luciferase, you can limit ATP, and then the ATP will be delivered by the cells and you monitor viability. And you can also limit the substrate, and then you would use a pro-substrate, which is converted to the substrate by the activity of a specific enzyme, so that in the end the light output of that reaction correlates with the activity of that enzyme. Our gold standard assay for determination of cell viability is the CellTiter-Glo assay, which is a luminescent assay that quantifies the amount of ATP in your sample. This assay also follows this add, mix, and measure principle, so we have a single reagent which contains all the required components. So this reagent contains all the components of the luciferase reaction except the ATP, and it contains a lytic component that will lead to cell lysis in order to release the cell's ATP, which will then drive the luciferase reaction, so that the light output will correlate with the amount of ATP, and from this you can conclude on the viability of your cells. An example for a cytotoxicity assay is the LDH-Release assay, in which we primarily monitor the integrity of the cellular membrane, because if that is lost, the cytosolic lactate dehydrogenase will be released from the cells into the supernatant, and by coupling now the LDH reaction to another enzymatic reductase reaction, which actually converts a substrate into luciferin, we can now correlate light output with the amount of LDH in the supernatant. This assay you can actually use in a kinetic fashion because it is so sensitive. You can collect tiny amounts of samples, so around 2 to 5 microliters at different time points, and you can either measure these samples directly or you can even store them at -20 degrees and perform the measurement when you collected all your samples. And this is some data that has been generated using that assay. So cells treated with a cytotoxic compound, and you can see a nice dose and time-dependent increase in the luminescent signal due to an increase in LDH release. Also, a hint when using such an assay regarding LDH release is to use heat-inactivated fetal calf serum because FCS is a considerable source of LDH, and by this you would simply raise your background and therefore decrease the sensitivity of the assay. Another factor that you should be aware of when performing cytotoxicity measurements is that cytotoxic markers have a time-dependent profile, and also there are markers which are really specific for a certain kind of cell death. So for example, caspase activity, which goes up very rapidly in apoptotic cell death, is completely absent in necrotic cell death. In contrast, LDH release is high at an early time point in necrotic cells dying by necrosis, and it appears very late in cells dying by apoptosis when these cells actually become secondary necrotic. And also levels of ATP change in a different manner. So in necrotic cells, ATP levels decline very rapidly, while this is more modest in apoptotic cells. So as you can see, the timing of your assay is an important factor. And this is further emphasized by this experiment where cells were treated with terfenadine, and 3 different parameters, ATP content, LDH release, and caspase receptor activity, were assayed. At an early time point, you can nicely see that we have an increase in cytotoxicity and decrease in viability. However, if we would perform the same experiment at a later time point, we in fact see a decrease in LDH release and a decrease in caspase activity. So that when using a toxic cytotoxicity marker that is activity-based, then we have the problem that this marker can get degraded or inactivated, and it's really a challenge to find the right time point to look at. And one strategy to approach this problem is to perform real-time measurements. And one opportunity you have is, for example, to use the CellTox screen cytotoxicity assay, which is based on a fluorescent DNA binding dye, which is membrane impermeable, so that in viable cells it will not enter the cell. However, if that cell loses the membrane integrity, it will enter the cell and bind to DNA and yield a fluorescent signal. So using that assay, you can actually perform real-time measurements of up to 72 hours. On the other hand, you also have the ability to perform real-time measurements looking at cell viability. And this is one example, the real-time-Glo MT cell viability assay, which is based on a different luciferase, the so-called nanolook luciferase, which converts furanocin into light. And in this assay, the luciferase as well as a pro version of furanocin is added to the cell culture supernatant, and in viable cells, the pro substrate is converted to furanocin and subsequently to light, while this process cannot take place in dead cells. And similar to that fluorescent cytotoxic assay, with this assay you can also perform real-time studies up to 72 hours in order then to identify really the time points that you want to look at more closely in future experiments. And with this, I'd like to close and hand over back to Jessica, who's going to tell you a little bit about cryptic contamination of your cell culture.
Thank you, Erik. So, in this last chapter, there are two types of contamination I would like to talk about here, because both of them are not so easy to detect and can have dramatic effects on your assays or other cell-based experiments. The first ones are Mycoplasma, a special type of bacteria, which are too small to be seen in a standard light microscope. They don't cause turbidity or color change of the culture medium like other bacteria and are therefore often overlooked. The other type of contamination that often goes undetected are the so-called eukaryotic contamination. These are cross-contaminated cell lines or misidentified cell lines. There are estimations that between 15 and 20% of cell lines used worldwide are either cross-contaminated or misidentified. I want to go a little bit deeper into the topic of how mycoplasma affect cellular metabolism by showing you some data from a study published in 2013. In this study, HeLa cells were infected with mycoplasma, followed by analysis of differential gene regulation up on different time points of infection. As you can see in this graph, the number of genes that respond to the mycoplasma infection increases dramatically with time. And I just want to briefly show you an extract of the list of genes that were identified. I know it's not very good to read. The cellular effects were dramatic. The genes that were deregulated upon infection are associated with signal transduction pathways, immune responses, cell cycle regulation, cell growth, and other cellular functions. And this is the reason why cell cultures need to be tested for mycoplasma on a regular basis. If they are tested positive for mycoplasma, the infected cultures should be discarded immediately to avoid spreading the infection. When you get cultures from another lab or when the status of a cell culture is not known because you have to wait for the test results, retain the potentially infected cultures in quarantine, for example, in a separate CO2 incubator. There are different ways to test for mycoplasma, every method that has its advantages and disadvantages. The best test method for you depends on different factors like the access to the required equipment, the number of samples you need to test, and the time you can wait for the results of the test. On our website, eppendorf.com/cellexperts, you can download a PDF file with an overview table listing the different mycoplasma test methods, their requirements, and their pros and cons. And this hopefully helps you to choose the best test method for your lab. Let's come to the eukaryotic contaminations. A misidentified cell line can develop from a cross-contamination of two different cell lines. Usually this happens by an accident somewhere during the handling of the cells. So it is a mistake in aseptic handling. Mislabeling of samples and inconsistent nomenclature of cell lines also contributes to this phenomenon. A cross-contamination can occur when you handle two cell lines at a time in the laminar flow. By accident, a little droplet of the more rapidly growing cell lines gets into the culture, shown here on this graphic in green. Because the pink cells are growing faster than the green ones with the following steps of cell culture, the pink cells become, you will end up with a dish of pink cells or a mixture of both. So as soon as two cell types are mixed up, it is very hard to distinguish those two. This process goes fast and it might go undetected. But although the phenomenon of cross-contaminated and misidentified cell lines has been recognized for half a century, as I mentioned before, still to date, we know that 15 to 20% of all cell lines used are either cross-contaminated or misidentified. Once misidentified cell lines are used for studies, the scientific as well as financial implications can be disastrous. Here are two examples of contaminated cell lines and their footprint in the scientific literature. Due to contamination in the 1960s, both are now known to be composed of HeLa cells. meaning scientists who thought they were working on HEP2 or IN407 were in fact likely experimenting on HeLa cells. Nearly 5,800 articles in more than 1,000 journals may have confused HEP2 with HeLa. More than 1,300 articles in 271 journals may have mixed up IN407 with HeLa. All in all, 7,000 plus publications have been cited roughly 14,000 times. It was calculated that more than $700 million have been spent on published work involving just these two contaminated cell lines, and $3.5 billion spent on subsequent work based on those papers. And these are just two examples. And again, I would like to get some feedback from you and start our next poll question. So, when do you perform cell line authentication? Again, please select all answers that apply. So, you can choose every time I get a new cell line into the lab, only when I receive cell lines from a non-reliable source, when establishing frozen cell stocks, prior to publication, or you can choose I do not authenticate cell lines. So again, I will wait a few seconds and give you time to answer. So we will wait a few seconds and then we will send the results and share the results with you. So I see that the first answers are coming in right now. Again, we wait a few more seconds. Okay, so the answers are continuing to come in. Okay, just a few seconds more and I'm going to show the results to you. Okay, so we have like 43, almost 43% of you is performing cell authentication every time they get a new cell line into the lab. Only 7% is performing it when they receive cell lines from a non-reliable source. 28% is authenticating the cells when establishing them from frozen cell stocks. No one is authenticating the cells prior to publication, and 20% does not authenticate their cell lines at all. Interesting. Okay, thank you for participating. So for those of you who want to know more about how to authenticate cell lines and check their identity, here is an overview of the method commonly used for authenticating human cell lines: short tandem repeat or STR profiling. The analysis of short tandem repeats has become the standard for intra-species identity testing of human cell lines. STR profiling can be performed in-house only if the required equipment is available, and adequate interpretation of the data requires quite high expertise and routine, so it can be more advisable to use a service provider for cell authentication. So we are almost at the end of the presentation. For more information on cell identity and reproducibility, please visit eppendorf.com/cellexpert. You can also find information about webinars and on-site training courses for different topics, including cell culture. We do one on-site training course here in Germany together with the colleagues from Promega. Before we come to the last slide, here comes the last poll question for today. We would like to ask you for some feedback, so please let us know, how do you rate the content of this webinar of today? So the questions you can click on are very good, good, okay, or poor. Thank you for your feedback. And yeah, that was it for today, for the presentation. And I think we will soon start the Q&A session now.
Thank you, Jessica and Erik, for your informative presentation. We will now start the live Q&A portion of the webinar. If you have a question you'd like to ask, please do so now. Just click on the Answer question box located on the far left of your screen, and we'll answer as many questions as we have time for. So let's get started. Our first question is, this one is going to be for Jessica. How do you define passage? Do you consider the freshly thawed cells as passage 0?
I increase the passage number every time that I trypsinize the cells. So when they are freshly thawed, I don't increase the passage number, but when I freeze them, I have to trypsinize them before to detach them from the surface, and then I would increase the passage number. So passage 0, not when they're freshly thawed, but yeah, every time the cells see trypsin or any other detachment solution, I would increase the passage number. But people do it differently, so the important thing is that you agree on one method in your lab so that everyone is treating the cells the same way with regards to the passage number.
And our next question is going to be for Erik. Are your assays also applicable to 3D culture?
Well, primarily our assays were designed for 2D culture. However, like the CellTiter-Glo assay, we also have in a 3D culture applicable version. And usually all lytic cells, the limiting step in using these assays for 3D culture like spheroids is the lysis of the cells. So you have to achieve a thorough lysis of your cells. And so it does not, you can adapt your assay to 3D culture by modulating that lysis step. For example, increasing lysis incubation times, or you could also mechanically enhance cell lysis. So this is something you can do. And for like the LDH-Glo assay, so where the lactate dehydrogenase release is measured, so such an assay where you actually use the supernatant of the cells, this is of course then also applicable to 3D culture, because you don't have to achieve any lysis of these cells.
Now, Jessica, if Mycoplasma contamination is discovered, would you recommend or discourage using a Mycoplasma elimination kit?
If you can replace the cells, I would always say it's better to discard them immediately and not use one of these elimination kits. However, when you use cells that are too valuable or that are irreplaceable for whatever reason, you can try to use these kits. However, I would always keep in mind that the treatment with these kits may also have an effect on the cells themselves. So I would monitor the cells very closely when using this kit and give them some time to recover and not use them instantly for an assay after you have done this this Mycoplasma elimination treatment.
Our next question is going to be for Erik. What happens if I use a black plate for luminescence and a white plate for fluorescent measurements?
Okay, so you could do that. So you can perform a luminescence measurement using a black plate. What you have to be aware of then, because a black plate just does not have this reflective properties such as a white plate, your absolute signal intensity will decrease. So usually around 1 log unit in magnitude. And if you do the other way around, if you perform a fluorescent measurement using a white plate, then of course, because you have these reflective properties, your values will be much higher with that plate. And also your background will be much higher because you miss that background decreasing effect of the black color of the black colored plastic.
We are getting so many good questions in today, so let's continue on with Jessica. Does the evaporation effect occur in our well plate sizes, for example, 6 well, 24 well, and 48 well?
The edge effect mainly occurs in 96 well plates or smaller well plates due to the small working volume. For plates with bigger wells and higher volumes, it has not been described yet as far as I know, but there might be differences depending on the cell type and experimental setup you are working with, and for this reason, you can fill the inter-well space of all Eppendorf cell culture plates no matter which size they are, and this way you can prevent any potential evaporation effect that might have an impact on your experimental data.
Now, Erik, do the substances you use in the real-time assays have any toxic effect on the cells?
No, they do not, at least. As I showed you, these assays can be used up to 72 hours. So we performed, of course, analysis for that time frame. For example, we added that fluorescent dye to cells at for different incubation times, and we then, for example, compared IC50 values, and we did not see any change of these values, so it really seems that these substances are non-toxic to the cells.
And it looks like we have time for one more question. So Erik, we're going to stay with you. What plate type do you recommend for multiplexing of a fluorescent and a luminescent assay?
Okay, well, as I said, for the second last question, you could do a fluorescent assay using a white plate also, and you could do a luminescent assay using a black plate. So if you combine a fluorescent and a luminescent assay, you pretty much could do it both ways. Generally, for our assays, we have a recommendation included in our technical manuals. However, a general rule could be that you look what signal is the weakest, and then you pick a plate. For example, if the luminescent signal is the weakest, you would go for a white plate, or the other way around.
Thank you again, Jessica and Erik. Do you have any final comments for our audience?
Well, thank you for joining us today, and thank you for your attention, and yeah, have a great day.
Yeah, also, I would like to say thank you, and if any further question comes up later on, feel free to contact us.
Before we go, I'd like to thank the audience for joining us today and for their interesting questions. Questions we did not have time for today and those submitted during the on-demand period will be addressed by the speaker via the contact information that you provided at the time of registration. We would like to thank Jessica Wagener and Erik Bonke for their time today and their important research. We would also like to thank Labyrinth and our sponsor, Eppendorf, for underwriting today's educational webcast. This webcast can be viewed on demand through May of 2019. Labyrinth will alert you via email when it's available for replay. We encourage you to share that email with your colleagues who may have missed today's live event. Until next time, goodbye.